Last time I described a type of cross performed by Mendel, more specifically the monohybrid type cross. And from that cross and that analysis of the data, phenotypes that he observed in F1 progeny and F2 progeny, he was able to derive fundamental laws of heredity that apply to all diploid sexually reproducing organisms. So again, the idea of diploidy, the idea of an allelic relationship, specifically of dominance and recessiveness for two alternative forms of expression of a given trait. And then lastly, the idea of segregation, the law of segregation, which again describes what occurs during a meiotic event, during gamete formation. Okay, so what I want to describe now is another type of cross that Mendel performed, from which he was able to derive additional information about the biological basis for heredity. So this is a dihybrid analysis. And so what he would do is he would, just like with the monohybrid crosses, he would choose two pure breeding parental types that contrasted in their form of expression for two of those 7 traits that he was analyzing in his studies. So for this particular example, we're looking at one strain, one parental type that was pure breeding for yellow round seeds. So the two traits, again, are color, seed color and seed shape. And then he had another parental type, another bag of seeds, a different strain that was pure breeding for green and wrinkled seeds. So alternative forms of expression of those two traits. So again, what he would do is he would plant these seeds, let them grow into plants, allow them to flower, and then he'd get out his paintbrush and effect a cross pollination or a cross fertilization event. And he would generate a hybrid seed, an F1 hybrid seed. And as you can see here, for this specific example, is that all of the hybrid seeds were yellow and round. And that's exactly what you would predict from his monohybrid analysis for each trait individually. So nothing too surprising there. So then he continued his analysis. And so just like with the monohybrid analysis, is that he allowed those F1 hybrid plants, he'd plant them, they would grow, they would flower, and he would allow them to self-fertilize to generate F2 progeny seed. And of those seed, what he saw was a new segregation pattern. And that segregation pattern, phenotypic segregation pattern. And that's what's being summarized down here. So he saw 4 phenotypic types among the F2 progeny in terms of the trait expression for seed color and seed shape. So you can see that one phenotypic type were yellow and round, another was yellow and wrinkled, a third was green and round, and then the last one, the fourth, was green and wrinkled. And the other thing about these 4 phenotypic classes is they segregated in a very characteristic pattern, a characteristic ratio. And the ratio is 9 to 3 to 3 to 1. Okay, so that's his observation from his dihybrid crosses. So what he could actually do is, we gave in this particular example, chose the 2 traits of seed color and seed shape. He could choose any 2 of those 7 traits that he was analyzing in his studies, and he could get the same segregation pattern in that F2 population for those 2 traits. There would be 4 phenotypic types, and they would always segregate in a ratio of 9 to 3 to 3 to 1. So that's the general observation that Mendel observed in terms of inheritance when you're following 2 traits simultaneously within a single set of crosses. So again, he was able to look at that and make some sense out of it and actually derive a 4th law of heredity. So here's his general observation. So again, in his F2 populations, for whichever 2 of the 7 traits that he chose, 9/16 would be dominant in their expression for each of the two traits. Another 3/16 of that F2 population would be dominant for the first trait and recessive for the second trait. Another 3/16 would be recessive for the first trait and dominant for the second trait. And then lastly, 1 out of every 16 in that F2 population would express the recessive form of the two traits. So this is the general observation. And from this general observation, again, he was able to derive a fourth law of heredity, and this is what we call the law of independent assortment. So again, something that I referred to previously when we were talking about meiosis. And so again, we'll get back to that in a little bit. So what did he mean by independent assortment, or what did Mendel conclude from those observations about this this law of independent assortment? What he said, or what he concluded, was that during gamete formation, segregating pairs of unit factors of heredity for two different traits, the alleles that exist for those two different genes, assort independently of each other into, the different gametes that are formed during gamete formation. Okay, so how did he come up with that idea from these observations here, this general observation of a 9:3 to 3:1 phenotypic segregation ratio? So this is what Mendel saw in this type of data. So if you analyze this F2 population for each trait individually, just like a monohybrid analysis, what you see is that if we're looking at color first, 12/16 of the population are yellow and 4/16 are green. Okay, so they're segregating 3:1, exactly as you'd expect for a monohybrid trait or a monohybrid cross in an F2 population. So if you look at the other trait, you look at round versus wrinkled, again, what you can see is that 12/16 are round and 4/16 are wrinkled. So again, that's a 3:1 segregation ratio for round to wrinkled, exactly what you'd expect from the monohybrid crosses that Mendel performed. But now we want to look at them together. We're doing a dihybrid analysis. We're looking at inheritance of the two traits simultaneously within a single cross. So this is where he came up with the idea of independent assortment. So of those that are yellow, that's these two here, that's 12/16 of the population, right? So what you see is that 9/12 of them are round and 3/12 are wrinkled. So of those that are yellow, 3/4 are round and 1/4 are wrinkled. Then you go over here, of the green seeded plants in that F2 population, that's 4/16 of the population. And what you see is that 3/4 of them are round and 1/4 are wrinkled. So what Mendel saw in this is that it didn't matter whether you inherited the yellow seed color or the green seed color, irrespective of which one of those two alternatives you inherit, you still segregate 3:1 for seed shape. So in other words, the two traits are being inherited independently of each other. So you can do it the other way. You can look at those that are round, that's 12/16 of the population, 9/12 are yellow, and 3/12 are green. So 3:1. Of the round seeded plants, they segregate 3:1 for color. So then you go down here, you look at the wrinkle seeded plants. That's 4/16 of the population. 3/4 are yellow and 1/4 are green. So again, it doesn't matter if you're round or wrinkled, whichever form of that particular trait that you inherit, you're still going to segregate 3:1 for color. 3/4 yellow and 1/4 green. So this is what Mendel saw in this data. This is where, he had the insight that very a few others at the time, could see what he was seeing in this data, and this is why he was a little bit ahead of his time. Okay, so here's another way of sort of summarizing what I just described. So again, if you look at each trait individually, in that F2 population of that dihybrid cross is 3/4 are yellow, 1/4 were green for the one trait, the color trait, and 3/4 are round and 1/4 are wrinkled for the shape trait. Okay, but now we want to look at them together. So again, what I described to you is that of those that are yellow, the 3/4 that are yellow, you still segregate 3-to-1 for round to wrinkled. And of those that are green, you know, the 1/4 of the plants that are green, that produce green seeds, they segregate 3-to-1 for round to wrinkled. So that is because of independent assortment, or that's what Mendel concluded. So what we're doing here is sort of giving you a prelude to a tool that's used by geneticists to predict outcomes of crosses when you have independent assortment, something called a branch diagram. But we'll talk more about that later. The idea here is, again, so if you're looking at each trait individually, so for instance, as I said, 3/4 of the plants in the F2 population are going to produce yellow seeds, 3/4 are going to produce round seeds. But what if you look at the two together? Well, of the 3/4 that are yellow, you would expect 3/4 to be round. So if you multiply 3/4 by 3/4, you get the 9/16. You know, the 9/16 that are yellow round, that are dominant. So of those that are yellow, again, 1/4 are going to be wrinkled. So 3/4 times 1/4, that would predict then that 3/16 would be both yellow and wrinkled. And then so on down the line, you know, so you do all of this multiplication in this branch diagram, you see a segregation ratio of 9 to 3 to 3 to 1 of dominant, recessive, dominant, recessive, just as this is the prediction, and this is actually what Mendel observed. So what I want to point out here is that, you know, what are we doing with this multiplication? So actually what you're doing is you're applying a law of probability, and more specifically, a product law of probability, which simply states that the probability of two independent events occurring simultaneously is simply the product of the individual probabilities. So it's the same idea as flipping coins. If you have two pennies, what's the probability that you will flip those two pennies, each flip being an independent event, and they will both land heads? So it would be a half times a half, which is a quarter. Or alternatively, what if they're both tails? You know, then again, it would be a half times a half is a quarter. So this is the same law of probability that we're applying here, because you have independent assortment, two independent events occurring simultaneously, the inheritance of the alleles for the two genes controlling the two traits. So again, 3/4 times 3/4, the probability of exhibiting that dominant phenotype for each individual trait. And together, because of independent assortment, the inheritance is independent of each other. You can multiply those values together to get the 9/16. Okay, so again, that's an application that is used within these branch diagrams, and we'll talk more about in a little bit. Okay, so again, from this dihybrid analysis, Mendel was able to derive a fourth law of heredity that is applicable for all diploid sexually reproducing organisms, as long as you have independent assortment. So I want to go through the same sort of analysis of a Mendelian dihybrid cross, just like I did with the monohybrids, but looking at it from a more modern day perspective, because we know that his unit factors of heredity, the genes and the alleles, are localized on chromosomes. We know how chromosomes behave through cell divisions, and in particular during meiotic cell divisions when gametes are produced. So again, using the same example as we've been discussing, we're going to define the seed color gene as being the G gene. There's two allelic alternatives, again, represented symbolically with an uppercase G being the yellow allele and the lowercase g being the green allele. And then we have the seed-shaped gene, we'll call it the W gene. Again, uppercase W is round, and a lowercase w is the wrinkled allele. So in Mendel's crosses, again, he started with pure breeding yellow round as one parental type and pure breeding green wrinkled as the other. So again, when we say pure breeding, what more specifically does that mean from a genetics perspective? It means that the individuals are homozygous. So in other words, for these phenotypic descriptions of the parents, we can now assign genotypes. So again, using the symbols within the model that I just described, this individual is going to be homozygous, big G, big W for pure breeding for yellow and round. This parent will be homozygous recessive for both G and W, the two recessive traits, green and wrinkled. So being homozygous during a meiotic event, during gamete formation, either of those two parental types can only produce one type of gamete. Again, when you reduce the diploid to the haploid, for this parent, one gametic type with containing one little g and one little w allele, and over here, one big G and one big W allele. Okay, so only one gametic type being produced by each of the two parents. So they fuse in a fertilization event, a seed develops, that seed represents the F1 hybrid. And what do we then know genotypically for that F1 hybrid offspring is that it can have only one genotype. It's doubly heterozygous for both G and W. So a dihybrid genotype. And what do we know, again, based on our model, as well as through Mendel's observations, this genotype, based on the model that I described earlier, should be yellow and round because of the presence of the big G and the big W. And that's actually what Mendel observed. All of the F1 hybrid seed, dihybrid seed that he produced from this cross were yellow and round. Okay, so that's pretty straightforward. But what about then now trying to predict the outcome of and a segregation pattern, both phenotypically as well as genotypically among F2 progeny? So what did we do with the monohybrid analysis in order to derive that information? We used a Punnett square. And to perform a Punnett square, what you have to determine first are the gametic types that the individual parents in the cross can produce. So again, through a selfing event, remember Mendel, these are pea plants with the bisexual flowers, so they, without any intervention, they will naturally self-fertilize. So through this self-fertilization event, will then generate the F2 progeny. So the question is from this dihybrid genotype on both the male and the female side, what are the gametic types that the individual can produce? We should be able to figure it out fairly straightforwardly. I mean, big G is going to segregate into a gamete, little g into another gamete. But when big G segregates into a particular gamete through meiosis, there's an equal probability of it segregating with either big W or little w. So when big G segregates into a gamete, because we have independent assortment, we already know that from Mendel's work, is that there's an equal likelihood of either big W or little w segregating with that big G allele. And then the alternative is that little g will segregate into a gamete, and then there would be an equal probability of either big W or little w segregating into the gamete as well. So you should be able to determine, based on independent assortment, that these are all four of the gametic types that the male and female parent in this selfing event can produce. And all four of these gametic types will occur with equal frequency. So a quarter like this, big G, big W, a quarter like this, a quarter like this, and a quarter, little g, little w. So then once you've determined that, you can assemble your, once you've determined the gametic types that the two parents can produce, you assemble your Punnett square. And in this case, it's going to be a 4x4 boxed array. Okay. And what do you do to predict the genotypic and phenotypic outcomes among the F2 progeny? Well, each of these boxes represents a potential fertilization event. Again, remember, fertilization events are assumed to be random. So if these two gametic types from, the big G, big W from one parent fuses with a big G, big W containing gamete from the second parent, this would be the outcome in the box, both genotypically as well as phenotypically for that offspring, for that F2 progeny. And you fill in all 16 boxes. Okay, so we'll get back to that in a second. I do want to point something out, this idea of independent assortment and trying to unify the two different ways that I've presented it to you, both from the Mendelian perspective as well as the perspective of, describing meiosis, and in particular, meiotic 1 metaphase, and then the subsequent anaphase. So what we told you is that, so we're, again, in this example, we're talking about F1 individuals that have a dihybrid genotype, heterozygous for G and heterozygous for W. So what I've done in the diagram is I've depicted metaphase 1 of meiosis for this dihybrid genotype. And what you need to understand is that for independent assortment to occur, the two genes have to be on different chromosomes. And so that's how I've drawn it. So you have one chromosome, the stippled chromosome that contains the big G allele, and then the white chromosome carrying the little g allele for the G gene on that one chromosome. So the W gene locus is on a second chromosome within the P genome. And this one, the stippled member of the homologous pair, carries the big W allele, and the white chromosome carries the little w allele. So with this particular alignment of these tetrads at the first meiotic metaphase, you're going to proceed through anaphase 1 and then meiosis 2. And so the end final resulting haploid products are represented here and over here. And you can see these are little g, little w containing. These are big G, big W containing. So those are the haploid gametic types that will result from this type of arrangement or alignment of these two tetrads at that first meiotic metaphase plate. But again, remember, there's another way that they can align relative to each other. And so that's what's shown here. So with this particular alignment, and then the subsequent segregation through meiosis 1 and meiosis 2, now you have two different gametic types that result. You can see over here, this is big G, little w containing, and over here is little g, big W containing. So from those two different alignments, possible alignments of the tetrads at that first meiotic metaphase, you can produce four different gametic types, all with equal probability or all with equal frequency. And again, that's what's summarized here, the slide that I showed you earlier. Okay, so again, once you've determined those gametic types, you can assemble your Punnett square. And here's the Punnett square. You fill in each of the individual boxes as representing a random fertilization event between gametes derived from the two different parents. You then predict the genotype and a phenotype for that particular outcome. And then what you can do is, this is just the raw data, the raw information. You can summarize it in terms of all of the different genotypes that are represented within that F2 population of the dihybrid cross, and the phenotypic outcomes as well. So if you look at all, there's 16 different boxes, so 16 different genotypes, but actually, it's not 16 different genotypes, there's actually 9 different genotypes, because some of them are represented more than once. So you actually see 9 different genotypes that occur within the progeny of that F2 population, and they occur in a ratio of 1 to 2 to 2 to 4 to 1 to 2 to 1 to 2 to 1. Okay, so that's the information that is derived from that Punnett square. 9 different genotypes occurring in that F2 population when you're looking at a dihybrid cross and you have independent assortment. And that's going to be true. It doesn't matter which 2 of the 7 traits that Mendel chose. I mean, this would be the same sort of prediction, same genotypic types and same genotypic ratios in that F2 population. So what about phenotypes? Again, we're dealing with dominant recessive alleles. So whenever you have a big G, you're going to have yellow seeds. So that's here and here. And whenever you have a big W, you're going to have round seeds. So again, these are 4 different genotypes in that F2 population that will produce yellow round seeds. These 2 genotypes will produce yellow wrinkled seeds. These 2 will produce green round seeds, and this 1 genotype will produce both green and wrinkled. So again, overall, you predict a phenotypic segregation ratio of 9 to 3 to 3 to 1, which is exactly what Mendel observed in his dihybrid crosses. Okay, so the question is similar to going back to when I described the monohybrid analysis, is that because of the allelic relationship of dominance and recessiveness, you can have multiple genotypes that produce the same phenotype. So again, 4 different genotypes here produce the same phenotype of yellow round. 2 different genotypes produce the same phenotype of yellow and wrinkled, and then so on. Same thing, 2 different genotypes here producing the phenotype of green and round because of the dominant recessive allelic relationships. So there are times when a geneticist wants to be able to determine the underlying genotype of a given individual that is exhibiting, dominant forms of expression for the trait or traits that are under analysis. So again, this is similar to what I discussed for the monohybrid crosses. So how would you answer that question? How can I determine if I chose an F2 plant that produced yellow round seeds, how could I determine which of these genotypes was represented in that particular individual? So what I would do is I would perform a cross. You could do the F3 analysis as well, but again, more generally or more typically, what is used is a test cross, crossing to a homozygous recessive individual. And then what you do is you look at the segregation patterns, the phenotypic segregation patterns among the progeny of the test cross, who will then allow you to distinguish between these 4 different genotypes. So for instance, if I chose an F2 plant that had yellow round seeds and it had the particular genotype of big G, big G, big W, what would I predict in terms of a segregation pattern among the test cross progeny? So here's the cross, and here's the predicted outcome. You're crossing 2 homozygotes, so there's only going to be 1 genotype resulting among the progeny, in this case, heterozygous for both G and W, and therefore, phenotypically, all the test cross progeny are going to be yellow round. There's only going to be 1 phenotype, no segregation. Okay, so if you saw that, if you perform this test cross and you saw that as a result, you would conclude that the genotype of that unknown F2 plant was homozygous big G, homozygous big W. Okay, so what if we chose an F2 plant that had this genotype? It was heterozygous, it was yellow round phenotypically, but it's homozygous big G and heterozygous for W. What would happen if I performed the test cross? Well, that's what's being represented here on the left side of this diagram. So there's the genotype, here's the cross, the test cross. Here's a sort of a mini Punnett square to predict the outcome of that cross. And what you would expect to see with that genotype is a segregation ratio of 1:1 for yellow. All of the seed are going to be yellow, and then they're going to segregate 1:1 for round or wrinkled. So what does that tell you? It tells you if you're not segregating for color, then the original plant, the unknown plant, must be homozygous big G, and because you're segregating 1:1 for shape, that individual must be heterozygous. Okay, so if you saw this particular segregation ratio, phenotypic segregation ratio among the test cross progeny, you would conclude this as being the specific genotype of that individual plant. So you can go back here and do the same for this genotype. Now, what if you're heterozygous for G, but homozygous dominant for W, homozygous for round? Well, here's the test cross over here. And again, here's the Punnett square that predicts the outcome. And in this case, what you would expect to see is no segregation for seed shape. So all of the seeds are round. The test cross plants or progeny plants all produce round seeds, but they segregate 1:1 for color. So that tells you that you're heterozygous for color, but you must be homozygous dominant for shape. So heterozygous G, homozygous big W. So a different segregation pattern. And then there's one other genotype. So what if you're doubly heterozygous, big G, big W, little w, and you perform a test cross of an F2 plant, a yellow rounded, round seeded F2 plant that had that particular genotype, what would be the predicted outcome among the test cross progeny? So here's the genotype, here's the cross, here's the Punnett square that predicts the outcome. So in this case, because you're heterozygous for both traits, you're going to segregate for both traits among the progeny of the test cross. And in addition, because they are being inherited independently of each other, you're going to generate a test cross segregation pattern for a dihybrid genotype of 1:1 to 1:1. 1/4 are going to be yellow round, 1/4 are going to be yellow wrinkled, 1/4 are going to be green round, and 1/4 are going to be green wrinkled. Okay, so you can see for all four of those genotypes, among the F2 plants that produce a single phenotype, you can distinguish between the genotypes based on the segregation patterns that are produced through a test cross. And that's the value, that's one of the values of being able to perform the test cross. It allows you to determine the underlying genotype or genetic constitution of an individual that is expressing the dominant form of one or more traits. Okay, so I'm going to stop there at this point. Mendel did continue on with more crosses, a trihybrid analysis, and we'll pick it up from there next time.